So when we get into this chapter about evolutionary medicine, the first thing that I want to point out is that there's a reason why we do this at the end of the semester. With evolutionary medicine, we have to use and apply all of the concepts and all of those experiences that you've gained through the different case studies and different examples that I've been using in class. We have to use all of that to start to understand this very interesting, this very applicable discipline of evolutionary medicine. Now, when we deal with evolutionary medicine is the perfect, almost quintessential example of complexity. The reason why is because we have an observation. Maybe that observation is that a group of animals or a specific human is suddenly showing us symptoms of some kind of disease agent, we think. So we do some research and we figure out that, yep, those symptoms are due to some kind of disease agent. Now let's start to figure out, what that disease agent is doing within the individual's body. Where did that disease agent come from? Is that disease agent related to other disease agents that we've seen? Does this have wider ramifications beyond the species that I'm observing? Oh my gosh. Very quickly, we have gone from the atomic level all the way up to the biosphere level. And so in the lecture slides, I've got that beautiful slide that shows how we would start to address what initially looks like a simple situation. I've got an organism showing me some symptoms. I should be able to figure this out. And yet, because I have to address every level in the levels of organization, because I have to bounce around between my levels of analysis, I have a very complex problem. Now, when we look at evolutionary medicine, I have been kind of introducing you to some of these concepts throughout the semester so that when we look at this discipline as its own separate identity, that we can understand how interrelated it is with everything else. And hopefully you have a sense of how complex it is. So even though in this chapter and throughout the semester we mostly stayed focused on humans, I don't want you to come away with the misconception that this is only applying to humans. When we look at evolutionary medicine, we could be dealing with all kinds of different organisms, from our agricultural animals like cows and sheep and horses, to our beloved pets like our cats and our dogs and our birds that live with us, to all of the different organisms that might be in our environment. Remember when we talked about the canine parvovirus, some of you have brought up and mentioned West Nile. And then in the lecture slides, we saw that the beluga whales can get coronaviruses. And while I know that whales sneeze and that whales can get sick, my brain always stops and goes, now, what do you do for a whale that has a cold? Like how does that happen? And how do they deal with that? And how is the virus transmitted in an aquatic environment where generally populations can stay very far away from each other, but are disease agents being moved on ocean currents the same way that they could be moved on wind currents that we see in the terrestrial environment? And then my brain just goes down a rabbit hole, and I come back many days later, having thought about all of these weird intricacies, because generally we just overlook life in the ocean and we don't really talk about it or focus on it. So evolutionary medicine goes beyond just humans, but a lot of times we stay focused on humans because that's where a lot of the research has been done, and let's just admit our implicit bias when it comes to humans. Now, I've been using this term disease agent quite a lot, and I want to stop and just revisit some of the concepts that we've talked about in relation to disease agents. So remember we talked about viruses earlier this semester, and viruses are going to be essentially protein capsules with some information inside, whether that information is DNA or RNA. Now, for the sake of this class, we stayed on the side of saying that viruses are not living. Viruses need other cells in order to make more of themselves. So they violate that characteristic of life, that living things can make more of themselves, can copy themselves in some way. Viruses struggle to develop or exchange genetic information to make a next generation that has genetically new combinations. Again, they're going to need some kind of agent to facilitate that. We're not going to have two viruses bounce into each other and suddenly start sharing information, produce a new virus that has a genetically new combination. So we've got a couple of the characteristics of life that viruses seem to violate. And so that's why we're going to stay on the side of saying viruses are non-living. But I do want to now introduce to you, since you've been through the evolutionary biology class, another layer of intricacy here is that some people who are researching and studying viruses say that viruses are living. So we still have that debate about how we're going to classify viruses, whether they're going to be non-living or living. Most scientists say non-living, so we're going to stick with that for this class. But I want to introduce you to the idea that some people out there might be referring to them as living entities. Why? Because they can accumulate changes. And like these other disease agents I'm going to put up on the board, they too can respond to selective pressure. We'll get into that in just a moment. So we've got viruses that are a disease agent. And now we get into the three domains of life. One of the most common domain of living species that can be a disease agent will be species in the domain bacteria. This is where we talk about E. coli and Staphylococcus, the bacterium that causes tuberculosis, the bacterium that causes tetanus. So bacteria are definitely considered a disease agent. When we talk about the domain archaea, we don't see that there are a lot of disease agents in the domain archaea yet, but this is a domain that we're still studying. I wouldn't be surprised if in the future when I'm teaching this class, I introduce archaea species as being disease agents as well. And then the domain eukarya, we get a lot of disease agents. So things like protists. And we already studied a protist this semester when we talked about malaria. Another disease agent kingdom within the domain eukarya will be fungi. And then some of the other disease agents will be parasites. And we talked about those as well. Now, generally, when we're talking about fungi and parasites and some of the organisms in the domain eukarya, not only are we talking about species that have eukaryotic cells with a nucleus and membrane-bound organelles, but we're also talking about species that are multicellular. So keep that in mind. Bacteria will be single-cellular. Viruses will be just a package. And some of our disease agents will be multicellular. The reason why all of this becomes important is because when we start to study how species acquire disease agents, what those disease agents do, and how we might treat or mitigate or cope with or even stop the spread of these disease agents, we have to understand all of those different aspects of the individual thing. If all I'm dealing with is a protein capsule with some genetic information inside, that might be easier to cope with than dealing with, say, a multicellular parasite, which might have a lot more genetic information. It might have a lot more strategies for dealing with what I do and responding to and coping to what I do. So we've got lots of different things that are considered disease agents, are causing symptoms and syndromes and problems that either lead to debilitating circumstances and or death for the individual organisms. So this is why we have to talk about individual organisms and their immune systems. Most multicellular organisms are going to have some kind of immune system that's going to fight against these disease agents. Now, the immune system is hugely complicated, and the video that I gave you very quickly glosses over, and as complicated as that video is, it is still an oversimplification of the immune system. Everybody's immune system is different. It's based on what you have been exposed to in your life. So your immune system is going to be different from mine based on all of the things you have been exposed to, the different proteins you have eaten, the different proteins that have been in your environment, all of the different stresses that have happened in your life, all of the different chemicals that you've been exposed to, thinking about BPAs and volatile organic compounds and arsenic, and going all the way back to the epigenetic case studies that we started to study, that we studied earlier this semester, where it wasn't just the chemical in the environment, but sometimes also other people's behavior that were breaking off methyl groups or adding methyl groups and changing how genes are expressed. And then we've got the straight up, what did you get from your mom and your dad? When the sperm and the egg came together, what kind of genetic combinations happened? So what did you start with? How have things changed over time based on your exposure? I mean, there are just so many variables. The immune system is hugely complex. What's beautiful and amazing is that we can get the immune system trained. We have figured out how to use vaccines. And in the lecture slides, I tell you about the different types of vaccines that we use, but essentially they're all the same goal. If I give my immune system a little bit of this thing, can I train it to recognize it? And then should it ever see it again, my immune system will immediately go out there and start attacking it, killing it, getting rid of it, whatever it needs to do to keep my whole body from succumbing to this disease agent. It is absolutely beautiful and brilliant. And it's also rare. There are a lot of viruses and bacteria and parasites and fungi that we don't have a way to treat. We can't vaccinate against it. We can't do anything. We can't work out how to train the immune system to say, whoa, I don't want this thing in this multicellular body. I need to get rid of it. So the fact that we have any, I think, is just stunning, stunning, stunning. So when you were a child, hopefully you had that whole sequence of vaccinations. And one of the things that's interesting is that when you're a kid, you get something like a tetanus shot, and that's going to protect you or train your immune system to recognize the tetanus bacterium. And that's one of those bacteria that hang out in our environment. And I always think of it as the rusty nail. You step on the rusty nail. If there's tetanus bacteria on the rusty nail, your body will get it, and there will be awful, potentially deadly situation that occurs because of that. Or if you get the tetanus shot, when you step on that rusty nail, then your body recognizes that this bacteria is not supposed to be here. It takes care of it, and you don't die. And you think about how many rusty things are in our environment, and how many, just all the things that we deal with and get involved in, and cuts and scrapes and scratches and entryways into our body that our bacteria could get in. And with this vaccine, we can train our immune system to protect us. But the thing is that you have to go back for booster shots. So about every 10 years, you have to go back and get the booster shot. And essentially what that booster shot is doing is reminding your body, hey, you remember this? You remember what this looks like? You remember what that feels like? Yeah, if you feel that, please kill it. Don't let it live. We need to get rid of that. And so with some of our vaccines, we find that there's a long time. Our immune system remembers it for a very long period of time. And with other things like tetanus, you have to go back fairly frequently to just give it a reminder. I don't know if you've thought back on elementary school, or if you've even thought about your times tables, or some of those things that we just take for granted that we learned when we were younger. Every so often you have to go back and re-remember them. 20 years from now, you may have to come back to your evolutionary biology concepts. Things like genetic drift and changes in gene frequency and, the Hardy-Weinberg model. In 20 years, you probably won't remember it. You'll have to give yourself an intellectual mental booster shot. Hey, remember what that is? Remember how we use that? Remember how we apply that? You may even do some calculations on P squared, Q squared, 2PQ, and figure all of that out again, just to remind your brain, oh yeah, that's right. That's how all this goes. So we do find with vaccines that we sometimes have to re-remind our immune systems to do this. Now, we're still in the early stages with the SARS-CoV-2 virus vaccines. First of all, it's amazing that we have these vaccines. It's a new type of vaccine based on RNA, so it's exciting to see how quickly that went from experimentation to application. Now, behind the scenes, RNA vaccines have been worked on for a long period of time. So they weren't actually all that new, but the idea that we could quickly bring them to market based on all that previous research is really quite remarkable. So with the SARS-CoV-2 vaccine, we don't know yet if we're going to need booster shots, how frequently we're going to need those reminders to our immune system, if we even need them at all. But some of this early research is showing that we may need to have reminders to our immune system. Look for that spike protein. That spike protein on the coronavirus, we don't want that. We want to make sure that you are always attacking and getting rid of any of the viruses should you see it. Should it infect any of the cells, get rid of those individual cells. We don't want more of this. So here we are with this really neat vaccine that came about fairly quickly and hopefully will give us some help in fighting off this disease agent. Now, if you own a pet, a cat or a dog, hopefully you're familiar with this concept of booster shots. You have to go in fairly frequently to get the rabies and distemper and the whole suite of vaccines for your pets. We also do it for ourselves. And this is what has led to this beautiful place that we're in as humans on the planet, where we can have such long lifespans. And not just long lifespans, but a majority of people can have lives without all of the debilitating things that were a result of some of those past viruses and disease agents. So in my family, I do have family members who had polio and are severely crippled because of it. And as they're aging and their body has experienced decades of dealing with the debilitation, all of the grinding of the bones on one another, and the way how the muscles have had to compensate, it just continues to be increasingly painful. So it was a struggle while they were younger, and then as they are aging, that accumulated chronic problem makes for a very, very, very challenging end of life. So they were too young to be able to be vaccinated. They missed that vaccination window. They got polio. And so now they have to suffer with that. But I got the polio vaccine. I didn't have to go through that. Hopefully you never have to go through that. We don't have to experience these debilitating things. I mean, if it doesn't kill you, it can leave you severely disabled and struggling for the rest of your life. And that could be 60 years, 70 years, 80 years. That's a lot. So science is always looking at, or scientists are always looking at, what's causing the problem? How can we fix or resolve this? How can we make this no longer a problem? And then that leads to the benefit of longer lifespans. Not only do we get longer lifespans, but so do our animals and our pets and the creatures that we surround ourselves with. And so evolutionary medicine has really, really, really been an interesting discipline to watch develop and then to watch the huge impacts that it has. Please make sure to watch the videos and all of those links. The reason why I include those videos is because I want you to have a clear sense of what the mechanism of action is. So we have causation. We have places where there is correlation, especially when dealing with the immune system and how the immune system is going to react, because not everybody's immune system is going to react the same way. So we have places where we have causation. We have places where we have correlation. And ultimately, we're moving across those levels of organization. We're bouncing across levels of analysis. Now, the other big concept here is we have to understand our role in putting selective pressure on disease agents. So I'll start with antibiotics. Antibiotics are wonderful. They're chemicals that are selectively damaging or killing bacterial cells. But there is some research that's showing that our antibiotics may be having an effect on our eukaryotic cells, whether that's a human or a cat or a dog or a cow or whatever we have given the antibiotic to. It doesn't seem that that chemical is just specific to bacteria. But this is really new research. So right now in this class, in the textbook, and our paradigm is going to be that antibiotics selectively kill bacteria. Great. So you take the antibiotic. You kill all of the bacteria that don't have a gene to resist. You are putting selective pressure for the bacteria to mutate, to develop a gene to survive. Bacteria, remember, are living entities, so they want to keep persisting from one generation to the next. They want to live. So if we use a chemical to kill them, then they're going to develop some way to defend it. There's a lot of pressure for them to develop some way to resist that. And this is where we see antibiotic resistance in all kinds of different bacteria, whether that's gonorrhea, whether that's staphylococcus or E. coli, or. Part of this is our own problem. We expose lots of bacteria to our antibiotics. Some of them are going to develop the gene to resist that. And then because they've got the gene to resist the chemical, they're the only ones that survive, so they pass that gene on to the next generation, to the next generation, to the next generation, and very quickly, your entire population of bacteria is resistant to that chemical. So then you develop a new chemical. Yay! You just put new selective pressure on the bacteria to develop a resistance to that new chemical. So only those that have a gene or have the ability to resist that will survive. They pass that on to the next generation, and the next generation, and the next generation, and very quickly your population is nothing but bacteria that are resistant to your new chemical. So you're clever, and you go and you make a new chemical. Bacteria now have selective pressure to develop a resistance to it, and what we see is that Red Queen hypothesis being played out. Essentially, our relationship with the bacteria stays the same. Now, granted, it looks like for a little while we win, and it looks for a little while they win, and it looks like for a little while we win, and it looks like for a little while they win, as we go back and forth. Ultimately, it's all the running to stay in the same place. We're still going back and forth in our relationship. And this is the bigger picture. This is the larger context that we tend to forget because, man, champagne and confetti from the ceiling every time we're able to start winning. But then we got to think about in the future what's going to happen. And it doesn't just happen with bacteria. This happens with protists and fungi as well. I really want to focus on fungi because fungal infections are one of the few things that doctors don't necessarily know to look for. They try bacteria, they try protists, but when it comes to fungal infections, we don't see them very commonly, but we are seeing an increase in fungal infections as we have started to shift and change our environment. And the patterns of temperature and precipitation are changing, which are changing the places where fungi are growing. And so we're seeing fungal infections in animals and people that otherwise never existed or were so incredibly rare you wouldn't see them, but now they're becoming more frequent. Talking specifically about some fungal infections that have been happening in dolphins and in the redwood forests. Only when you go way out in the middle of nowhere, though, the redwood forests up in Washington, Oregon, and throughout Canada as well. And so that is in response to how we have changed and shifted the larger climate across our planet, and then individual habitats as well. And so when we use chemicals to try and kill the fungus, then the fungi can rapidly respond to that within just a few generations, very similar to bacteria. And so it's a very scary thing because quickly we see a future where we can't use antibiotics anymore because all of the bacteria, they all have genes to resist all of our chemicals. We can't use fungicides to kill the fungi. All of our chemicals are useless because we have been exerting selective pressure on those populations to develop genes and develop adaptations to resist the chemicals. And so this is a more concerning context of evolutionary medicine. We can see how to fix a problem. But what if through fixing the problem, we make the problem worse? And that's really where we're stuck right now, is that we can see where we've got bigger problems that we've created because of the Red Queen hypothesis, because we're locked in these relationships. And so the question is, how are we going to cope with this? And we don't have a good answer yet. It's probably going to require paradigm shifts. It's going to require thinking from a different perspective, thinking from this whole scale biosphere perspective. And that's really hard. It's really challenging because all I want to do is I want to make people feel better. I want to fix this problem. I want to fix dengue. I want to fix Ebola. But this is why I give you the case studies, is I want you to see how the disease agents work, what they do, how individual genetics can play a role in individual immune systems, and the individual and all of their variants can make a difference. I want you to start thinking at the population level, and then I want you to start thinking across the whole planet. I want you to grapple across all of these scales in a very dynamic way now that you have the background and the basics of evolutionary biology behind you, all of those concepts, all of the vocab words, and all of those previous case studies that we've done and previous examples that you can bring to play when you look at the Ebola virus, when you look at the dengue virus, and these different solutions that we have developed. So at this point, we're starting to fall off the ledge into some very, very, very high level thinking and very high level problems, and we're starting to get into higher level classes. And that's, of course, where I want to leave you, because I want to leave you prepared and ready, hopefully more informed, have a better understanding of the life around you, a better understanding of your relationship in this larger planet, in this larger biosphere and all of the different ecosystems. I hope you feel more knowledgeable, more empowered, and you have a better understanding of the planet around you. Congratulations on making it to the end of this semester. As always, email me. Never hesitate to come back and ask me more questions as they occur to you over time. And never stop being curious. Thank you for taking the evolutionary biology class.